Hello, my name is Jordi Diaz-Manera and I am Professor of Neuromuscular Diseases at Newcastle University. In this video I'm going to talk you a bit about MYO-Guide, which is a project where we are applying artificial intelligence to the analysis of muscle MRI for the diagnosis of patients with neuromuscular diseases. So muscle diseases are characterized by a degeneration of the skeletal muscles causing progressive weakness affecting mainly the proximal muscles of the limbs. These diseases can be genetic or acquired, affect both males and females, and can onset at any age from childhood to the adulthood. Examples of these diseases are Duchenne, Becker, limb-girdle muscular dystrophy, Pompe disease, or many others. There are more than 200 different types of muscular diseases, and each subtype represents a unique compilation of different signs and symptoms, and there is a significant heterogeneity between the different subtypes of diseases. So the diagnosis process of muscular diseases has changed considerably in the last 20 years. In the Sanger era, so this is before we had the revolution of the next generation sequencing, where genes were sequenced one by one, the diagnosis process started with the clinical history and clinical examination to the patient. Then we were performing different complementary tests, like for example muscle biopsies, MRI or electromyography, to identify the more plausible or the more potential cause of the disease, the gene, the candidate gene, and then we were doing the sequencing of that gene. If we identified a mutation, the diagnosis was closed. If we didn't identify a mutation, the diagnosis was still open, and we needed to repeat some test or going back to the first steps, going back to the clinical history and examination. Then this means that the whole process could be very long, and in some cases we needed a lot of time to reach a diagnosis, and in many cases we didn't reach a diagnosis of the patients. In the last years, the first decade of this century, there was a revolution in the way how genetic diagnosis was performed. The main reason is because they started to appear new techniques that allow to sequence many genes at the same time, what we call next generation sequencing, and this consisted in panels or exomes, where we could, for example, sequence 80 or 100, 300 genes involved or related with neuromuscular diseases, or also all the genes of the body. So this has clearly changed the way how we diagnose patients today. We start with the clinical history as in the past. We then do the clinical examination to have an idea of if the patient has a myopathy or have a neuropathy or have a congenital myasthenic syndrome, and then we do the panel of genes. This means that in many cases we end the diagnosis just after the panel of genes, but unfortunately there are some limitations of next generation sequencing. For example, next generation sequencing studies that not detect all types of genetic changes. There are some changes that are not detected using the conventional studies. Some diseases cannot be identified, and this is the case of FSHD, for example, or myotonic dystrophy. It is only useful for genetic diseases, of course, so if you have a patient that have an acquired disorder, you will never reach that diagnosis using NGS. It can identify variants of unknown significance, and that's a problem because then we need to validate the pathology or the pathogenicity of the new variant that has been identified. And NGS is not available in all hospitals. It's only available in a minority of countries. There are many countries around the world that they still use Sanger for the diagnosis of their patients. So the reality is that in front of this situation, complementary tests are still useful for the diagnosis. So one of the complementary tests that we use is muscle MRI. So the muscle MRI have been used for many years in the diagnosis process of patients with neuromuscular diseases because there are some changes that can be helpful. And here what we try to do is to collect as many MRIs as possible from patients with different neuromuscular diseases in order to develop a software using artificial intelligence that can be useful for clinicians. So the problem is that we have been doing MRIs for more than 20 or 30 years, and sometimes finding the patients to ask them to sign a consent form for collecting these MRIs, it's complicated. So the idea was to collect for this project non-identifiable data of the patients. So these are just the imagings of the MRIs. You will see some examples now. Some data, some personal data, like for example age, that can help in the diagnosis, gender, that can also help in the diagnosis, and also the diagnosis, just the name of the gene that is mutated in all patients, in order to develop this software. So just going with a bit of introduction to the MRI. This that you see in the screen, it's an MRI of patients with neuromuscular diseases. And why are we using MRI for the diagnosis? We're using MRI because there are some changes that are characteristic of different diseases, and that the changes are not random. So we know, for example, that if you have mutations in the gene A, for example, it can happen that you have a predominant involvement of the posterior muscles of the thighs, while if you have a mutation in the gene B, then the changes will be mainly in the anterior compartment of the thighs. So these changes that are not random and are dependent of the gene that is mutated have led to the description of the characteristic patterns of muscle MRI involvement in many different disorders. So the reality is that MRI was first described in the 60s. In the 80s, there are the first publications applying MRI to the study of neuromuscular diseases. And from the 80s to today, there have been many descriptions, many papers describing the MRI features of different neuromuscular diseases. And for some years, we have been using algorithms based in MRI to provide a diagnosis to patients with neuromuscular diseases, just focusing in the muscles that were affected and the muscles that were spared in each of these diseases. But the reality is that these studies have several limitations. Why? Because there are many patterns that have been described, and the reality is that the higher the number of patterns that are described, the lower the specificity of these patterns. We have seen that there are only a few diseases with characteristic or specific patterns. Many of these, in theory, characteristic patterns of MRI involvement are overlapping. And the reality is that for neurologists, a high degree of specialization is needed in order to recognize patterns that are suggestive of a diagnosis. So, in front of this situation, we asked ourselves some years ago, is there any software that can help? And the answer was no. And we decided to develop a software that was going to use artificial intelligence in the analysis of the MRI data in order to guide the diagnosis. At that moment, that project had three different objectives. The first one was to collect a high number of MRIs from patients with 10 different muscular dystrophies with a genetic diagnosis, in order to have a large collection on MRIs of these patients to work with. The second was to create a software that could apply artificial intelligence and develop a model that could be useful for the analysis of these MRIs. And the third one was to test this software with a new set of MRI to know the efficiency of the model. So these are the diseases that we work with in the first project. So as you can see, these are muscular dystrophies, some of them very well known, like dystrophinopathies. There are also some limb-girdle muscular dystrophies, calpine, dysferlin, sarcoglycans, FKRP, ANO5, and other diseases that can mimic some of the clinical features of patients with limb-girdle, like for example oculopharyngeal muscular dystrophies, laminopathies, FSHD, or also Pompe disease. So thanks to our collaborations, we were able to collect close to 1,976 MRIs from patients with a genetic diagnosis, and we started working with them. So we developed a software that was analyzing all the patterns, all the muscles that were affected in these diseases and the ones that were spared, and that was able to provide diagnosis, to predict the diagnosis based on the MRI data of these 10 neuromuscular diseases with a very high accuracy, as you can see here on the screen, higher than 95%, high sensitivity and high specificity. And this first software was called MYO-Guide and was published in the journal Neurology in 2020. So we have now moved into a new project that what we want to do now is to implement this module, this software, into a web page and open this to all the medical community around the world to facilitate the diagnosis of patients with neuromuscular diseases not only in the UK, also in Europe, also in America, also in countries where next generation sequencing is not available. So we want to increase the number of muscle diseases. So we have started with 10. We want now to go to 25 or even more. We would like also to develop an automatic segmentation tool, so a tool that can help clinicians to automatically identify the muscles that are affected and the muscles that are spared. And the third part is, of course, to create a web that can host this MYO-Guide, new version of MYO-Guide. So this is just a list of the new diseases that we are including. So we are including many individual, but we are also including other diseases that are affecting adult patients with neuromuscular diseases. You see myotonic dystrophy, real one myofibrillar myopathy, and also spinal muscle atrophy. We are also interested in increasing also the number of congenital myopathies, and there are some diseases here that are not genetic, that are acquired. In order to increase the number of diseases, as you can imagine, what we need is a larger number of MRIs. And for this reason, we have expanded the number of collaborators that we have in this project. So we started with just 5 collaborators, but you can see here in the map that we have now collaborators in Asia, we have collaborators in North America, South America, and also in many countries in Europe. And the way how these centers are sending their MRIs to us is using this platform that is known as MYO-Share. So MYO-Share is an online platform, imaging platform that is built in the Microsoft system Azure, that was developed by Jody Warman in the University of Ottawa. It's a pretty easy platform that allows to upload anonymized MRI and share securely MRIs between different investigators for a research project. So this is just a snapshot of MYO-Share, and as you can see here, what you have in MYO-Share is your patients, so your MRIs, the MRI that you have uploaded. You have the MRIs that you have shared with someone, for example, for a project, or the MRIs that you have also received. And this is another snapshot of MYO-Share, where you can see basically how the MRIs are seen. So the MRIs are here. We can also score the amount of fat using the Mercuri score here. We can download our scores, etc. So in this project, what we are doing is to collect all the MRIs through MYO-Share. We are scoring the amount of fat present in the different muscles in order to have the muscles that are more affected, the muscles that are less affected, and we are downloading these scores in tables. You can see these Excel files where we have, for example, this is one patient where we have a code, as you can see, is completely anonymous, the age of the patient, the gender, the diagnosis, and the Mercuri score for all muscles of the lower limbs, pelvic, thighs, and legs. So this is the information that we have. This is the information that we are using to build our model. So one of the important points here is that in order to be able to collect all this information without asking patients to sign a consent form, we need an approval of CAG. So CAG means Confidentially Advisory Group, and this is an organization that supports studies when taking consent from patients is not feasible. So CAG is an independent body that provides expert advice on using confidential patient information. They protect and promote the interests of patients and the public, and this mainly regulates the possibility to access confidential patient information without consent and without a breach of the common law duty of confidentiality. CAG gave us their support to do so, among others, because we are just collecting essential data in this study. As I have said, we are just collecting the diagnosis, the sex, and the age, and we are using it for the set-out purposes. And the main reason why we contacted CAG, as I said, is because we have MRIs of patients that were obtained maybe in 1995, and coming back to those patients it's very difficult, almost impossible sometimes. We have tried, but it's very difficult to reach them. And the second part is because what we are collecting is not really identifying anyone and can ease, facilitate, accelerate our research. So if you want to obtain support from CAG, you need to contact them, and you need to also provide your project. They will review it. They will see that this is not intrusive research, it's not an educational project. They will review the project, and they will ensure that the confidentiality of the patients is preserved. And then what they are going to do is to train the staff that is also participated in the project, to be sure that they are aware of the data that they can obtain and cannot obtain. And they also need to receive an annual report with all the MRIs that have been reviewed, with all the data that you have collected. And also they also want to know the opinion of the patient. So one of the reasons why we are doing this video is because we want you to tell us if you are happy if we collect the information about your MRIs for this project without requesting your consent. So the other important point is that CAC asks you to do poster and leaflets, and leave that in relevant departments of NHS premises. So we have, for example, this poster that you can see here, and this is something that can be seen in the clinics here in Newcastle and also in other sites. And it's basically what we do. So what is our experience so far in this new project? So what we have collected is we have gone to different hospitals in UK. This is just experience in Salford. As you can see, we went to Salford and we collected anonymized MRIs there, 182 MRIs of many patients with different diseases, as you can see, and we uploaded these MRIs into MYO-Guide to have them here in Newcastle and to analyze them here. The last part of the talk is about the web. As I mentioned before, we wanted to build a web to put our MYO-Guide project, and this is the web, is the www.myoguide.org. And here you can see that there is information about the people that are participating in the project, how to contact us, but I would like to mention this diagnosis part. So if you click diagnosis, you will enter into a secure part of the web where you need a username and password, and this is mainly for clinicians that want to use the software. How does this work? So they enter here, so they have an MRI, imagine that they have an MRI of a new patient, and they calculate the mercury score, so they calculate the amount of fat that is present in the different muscles of the legs. So they need to put here the different mercury score per each muscle. You can see a list of the muscles here, and therefore they will have something like this, for example, where you can see that there is a predominant involvement of the posterior muscles of the thigh in this patient. And once they have included all the muscles in the list, they press the submit button, and then they will obtain a distribution, a prediction of the disease based on the muscles that have been included. In this case, they can see that 51% chances of the patients to be to have a mutation in the DMPK gene. And so this is just a list of the current collaborators that we have. We would like to thank them for sharing the MRIs with us, because this has also helped us to improve the algorithm. In light blue you can see other countries that have been contacted, and we are aiming to expand our collaborators to many different countries. And this is just the people that are working in Newcastle in this project. I would like to thank the whole team. I would like to thank you for your attention, and also to all the collaborating sites in UK that are helping us to collect all the MRIs, and also to our funders. So we would like you to spend 5 minutes now and go to the following link. You can also have it here in YouTube. You can just press the button, and this will give you access to the form. And we would like you to fill this form and let us know if you are happy with not having a consent form signed for us to collect your MRI and use them in this project. Thank you very much for your attention.